Well, hello. I wanted to real quick offer you a little insight into mutation. So I understand that there was what am I trying to say, video on this, but I also wanted to just give a little bit more insight. So at its heart, I think it's important to understand that mutation is a key part of cellular replication. In fact, mutation is necessary to drive evolution. And in organisms that do not sexually reproduce, mutation is one of the biggest drivers of change from generation to generation. And that's important because it means that mutation can basically fall into three categories. It can be good, it can be bad, or it can be irrelevant. Okay? So what we're going to do today is we're going to talk a little bit about some of the ways that mutation happens, all right? Now, first, what we're going to do is we're going to focus on what is referred to as a gene mutation. So here we're looking at mutations to individual nucleotides, right? So let's take a look at the first type, which is referred to as a substitution. Now, a substitution mutation is when we have a situation where one, what am I trying to say? Where one base is swapped in for a different base, okay? So you can see up here you have this DNA strand that's basically just CAT over and over again. But what I have is right here, this A is changed for a C. And so this amino acid here, which was histidine, is now a different amino acid. I believe that says proline, right? And so what happens is now I have a new strand of amino acids. Now, this, which is called a missense mutation, could be good. It could drive change. It could make the protein work better or do something different that's valuable. Or it could create a problem where suddenly this protein does not work the way it is supposed to. And so this could be good, could be bad. All right. Now over here we have what's called a nonsense mutation. Now it's the same thing. I've still got this same nucleotide right here, but it is changed to something different this time. This time it's changed to a T. And what happened here is it inserted a stop codon. And so what that means is the remainder of this amino acid sequence is gone. Doesn't exist anymore. And so that is what's referred to as a nonsense mutation. Nonsense mutations are almost universally bad, right? Because they have a protein where huge chunks of it frequently are just gone. They don't work anymore. And since structure equals function, too much of the structure of that protein has probably been altered for that to work well. Now, finally, down here, we have another type referred to as a silent mutation. So you see right here, you have this GAA gives us the amino acid glutamic acid. Well, this A is mutated to a G, and now instead of GAA, I have GGAG. But look, it still gives me glutamic acid. And so that's a silent mutation because even though there is a mutation, there is a change to the nucleotides. There's no change to the amino acid. And so the protein remains unchanged. And so those are the three main types of substitution mutations: missense, nonsense, and silent. Silent mutations cause no change. Nonsense mutations typically are very bad, break down the protein. Missense mutations have the possibility to be good. They might drive evolution, but they also could be bad. Now, there is another type of mutation that is known as a frame shift. Now, here it's important to remember that our nucleotide sequence in DNA is read by transcription and translation in groups of 3. And so those groups of 3 begin at the first start codon. And then they work in groups of 3. So if I either insert an extra nucleotide, so you'll notice right here I have a C, and what's happened is an A has been placed right in front of that C, Or I delete a nucleotide. So here I have an A, which has been removed, and so that T just sort of shifts backwards. What that does is in either of those situations, I change the groups of 3 for everything after the mutation. So you look down here, it's CAT, 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 but down here it's CAT, 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 ACA, TCA, TCA, TCA. So everything after this extra nucleotide is inserted is now different, which means every amino acid is now different. That causes drastic changes to the structure of the protein, and consequently, drastic changes to the function. We have the same problem over here. I have CAT, 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 CAT. Down here I have CAT, 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 and then where that A was deleted, I now have CTC. And then I have ATC, ATC, ATC. So once again, every nucleotide after that deleted one is read in different groups of 3. And when that happens, all of a sudden every amino acid is now different. And that causes drastic changes to proteins, drastic changes to their structure, drastic changes to their function, and frequently those lead to the death of the cell. Now over here we have another thing called chromosomal mutations. Now chromosomal mutations can be the result, it's hard to be right. Weird. All right, that's not working very well. So they can be the result of either replication gone wrong. So you look at something here like duplication, right? That's something that was replicated more times than it should have been. Deletion, something was replicated and not replicated like it was supposed to be, right? Or frequently it could be a problem with crossing over gone wrong. And so that's when you see things like inversion and translocation, where pieces of whole chunks of the chromosome are put in different places. So let's talk a little bit about why chromosomal mutations are so bad. Well, for one, each chromosome is one long strand of DNA, literally billions of nucleotides. And so if you change chunks of that strand, you're changing millions of nucleotides at once. Whereas what we just talked about, we changed one nucleotide. And so chromosomal mutations are far more drastic, okay? When you delete chunks of a chromosome, you potentially do not produce protein. And you need your proteins, whether they're enzymes, structural, whatever. If you don't make them, your cells can't do their job. You can't live. If you overproduce a protein, which is what can happen with things like duplication, inversion, or translocation, you end up making too much of a substance you need. And since our bodies struggle to maintain homeostasis, which is a balanced internal environment, too much of a protein can also be a problem. Okay? So real quick, the deletion, a segment right here, I believe it's the segment that's D, has been removed. Okay. Over here I've got duplication. You can see this goes A, B, C, D. Here it goes A, B, C, B. So B has been duplicated, C has been duplicated. Okay. So you just have extra of that. Not good. Too much protein. Down here you have an inversion. So it goes A, B, C, D, E, but now it goes A, D, C, B, E. This generally is a problem because it changes the order of the nucleic acid, of the nucleotides. There's a potential for it not to cause a problem, but it will change the amount that your genes are produced. It'll change sometimes it'll actually alter the proteins. So inversions can be pretty drastic in their alterations as well, although they can be subtle. Now down here we have translocation, which is real drastic. So I've got one chromosome right here, and I have a different chromosome right here. And what's happening is these pieces are ending up down here, and these pieces are ending up here. That's a big problem. You don't want want that entire chunk of DNA to be with the wrong rest of the DNA. It can change, again, how much protein is produced, whether the protein is produced, the structure of the amino acids, of the proteins made, where those nucleotides meet. Chromosomal mutations lead to very drastic changes, and most of them will result in the organism being unable to survive. So there you have mutation. And the basics of it boil down to it is changes to your DNA. They could be small on the nucleotide level, which is what we saw with the first one, or they could be large on the chromosomal level, which is what we're looking at here. Some of those changes can lead to positive results. Some of them can lead to results that don't matter. Many of them will lead to results that stop a cell from surviving. All right, cool. That's mutation.